What is a cancer model? Basically, a cancer model allows researchers to study live cancer cells outside the human body. One type of model is called a cell line, which consists of immortalized cancer cells growing in a lab dish. Some other model types are mice, genetically engineered to have cancer, or that have human cancer cells growing under their skin. Researchers use these models to study cancer biology and develop new treatments in the laboratory. Today, we're going to focus on cell lines derived from patient tumor cells by touring a lab at the University of Colorado that makes and studies cell lines.
So here is our tissue culture room, and what we have here are tissue culture incubators where we maintain live cells. They're maintained at 37 degrees at a constant CO2 percentage to maintain the cells and keep them happy, actually. Here's an example of one of our cell lines that we use. Actually, it's a ROS1 cell line that we have derived from a patient.
Oops, got a glare. There we go.
And here's a microscopic view of this actual cell line.
This is how a ROS1 cell line looks under a microscope. You're actually looking at clumps of living ROS1 cancer cells. The little round spots are cell nuclei.
So we have the cells in culture, and once we need to do experiments, we transfer them to plates that are defined for the experiment that we need. In this case, I have a same cell line that I put it in these wells, and each plate was treated with different concentrations of 4 TKIs, 4 different TKIs. And I go from really high concentration to really low, and then once I treat it, I'm going to extract the proteins of the cells. So I put a solution that will dissolve the cells, and I'll collect that liquid to run and look for signaling proteins to see if the targets are being inhibited by the TKIs.
This graphic is called a Western blot. Researchers use it to detect protein signaling. This particular blot is looking at signaling from crizotinib-resistant ROS1 cell lines treated with different TKI combinations. Each set of colored bars represents a different concentration of a TKI or a combination of TKIs that have been applied to living ROS1 cells. If a ROS1 cell is signaling, it is not being inhibited. Green bars indicate the TKI is effectively inhibiting ROS1 cancer signaling, and therefore might be an effective ROS1 treatment. Lighter colored bars indicate the TKI is not effectively inhibiting ROS1 cancer signaling and is therefore not an effective treatment. The 4 sets of bars on the far left show crizotinib-resistant ROS1 cells treated with crizotinib alone. The concentration of the TKI increases as you move from left to right. Not surprisingly, the first 3 bars on the left show typical doses of crizotinib are not inhibiting the cancer. However, the 4th set of bars from the left is green, which means that concentration of crizotinib is effectively inhibiting the cancer. Unfortunately, that concentration is too high to be tolerable in humans because it would kill healthy cells. Further to the right are bars representing TKI combinations. Some of the bars are green, which indicates those TKI combinations might work against crizotinib-resistant ROS1 cells. With additional research and funding, such experiments might eventually lead researchers to develop new ROS1 treatments that are both effective and tolerable. We hope you've enjoyed touring the Doebele lab at the University of Colorado and learning about ROS1 cell lines and how researchers use them. Thank you to those who made this video possible.